meeting. In the technology sector, it's often celebrated that a few people can make huge breakthroughs. And sometimes people think that may be the way to get advances in all areas of science, including medicine. They have an image of breakthroughs being made just in the lab. And that work is very important. And that model has done great things. But today we're going to talk about advances that require hundreds of people and massive collaboration. We started Grand Challenges 16 years ago with that idea in mind, to build a global network of researchers, scientists, and innovators, and to identify the most important problems so that we could focus their collective brain power on these problems. Now, so many scientists all over the planet, whether they're in this meeting or not, are involved in a particular grand challenge, which is ending the current pandemic. All over the world, some of these researchers are breaking down silos. Rather than waiting to go through a publication process, they're sharing data on a daily basis. Since the pandemic began, scientists have shared more than 137,000 viral COVID-19 genomic sequences. The pharmaceutical companies are cooperating on production in ways that really have never been seen before. For example, the antiviral remdesivir created by Gilead is being produced in Pfizer factories. And international teams of scientists are collaborating at full speed on the vaccine clinical trials. Likely, one or more of these vaccines will be available by early next year. In fact, as we have multiple vaccines, we'll have to make sure we understand exactly where to use each of them in the time when those volumes will be constrained. So the pace of the science has been remarkable. And my bet is that when we look back on the history of COVID-19, one of the bright things will be that the scientific community worked together to come out with the tools to bring this to an end. But despite all this good work, right now, as fast as the science has moved and as great as this collaboration is, the pandemic is still ahead of us. The first COVID-19 vaccine will probably be the fastest that humans have ever gone from identifying a new disease to being able to immunize against it. Still, we all know this virus has managed to plunge the entire global economy into a deep recession, infect almost every country, and kill over 1 million people. As we hopefully enter the final chapter of this pandemic, we have to start asking, how can we do better before the next one? Where can we push the frontiers of science so that the next time a novel pathogen appears, it doesn't create a tragedy of historic proportions? The good news here is that COVID-19 has been an instructive adversary. It's pointing us to many areas where we need better R&D, and I want to identify 3 of those. The first is vaccine platforms that can let us develop vaccines many months faster than traditional methods. Imagine how this pandemic would have been if we'd had a COVID-19 vaccine by the summer of 2020. Since the outbreak of the virus, researchers have made huge strides in advancing some new platforms, but they still face time delays and barriers to scale and cost. First, let me take RNA vaccines. This is an area that I know many on the call, including the foundation, have seen great promise in and have been backing for many years. For the first time, they're being reduced to practice during this pandemic. Probably the first approved vaccines for COVID-19 will be mRNA, and yet we can't count on them alone because they're very hard to scale that manufacturing, and they have a logistical problem because they require an ultra cold chain. Scaling these for the world would mean having hundreds of millions of doses in liquid nitrogen and keeping them at that negative 80 degrees Celsius as they're carried from planes out to get delivered in the world. I'm hopeful that as the mRNA platform matures in the years ahead, we'll solve both the scaling problems, which will bring the cost down, and will make this cold chain requirement unnecessary. The Grand Challenges program has been looking at ways to make vaccines more thermally stable since the very beginning in 2003. And by taking advantage of that research and some new ideas, I think we'll be able to develop mRNA vaccines that don't require that ultra cold chain. Another promising area for rapid vaccine development is structured guided immunogen design. Here we have a different problem. Uh, we can design proteins with atomic level accuracy, and that gets us very quickly towards a subunit vaccine. But the subunit vaccines almost always require an adjuvant to induce the desired immune response. And this has become a major barrier, a barrier in cost and scale as well. We have very few approved adjuvants in the world, and not enough to make the billions of doses we need to stop this pandemic. Partly that's because the ingredients in the current adjuvants are so rare. Going forward, we need a way to make even more powerful adjuvants that don't require someone going to the high Andes to harvest the bark of a particular tree or shark fishing. Right now we use shark oil to make the adjuvants like squalene. Chemically synthesized adjuvants, ones that we can make without these restricted raw natural resources, need to be a solution here. We also need to develop small molecule adjuvants that act through different pathways of innate immune activation. These will offer a broad range of options for driving the type and duration of immune response, which we really need for the difficult vaccine challenges that we've been working on for a long time, specifically HIV, tuberculosis, and malaria. So more risk is required to advance new adjuvants, and I do think we can make huge progress on this in the next 5 years. The second area that's very important is monoclonal antibodies. This is an area where we also need more research. Platforms for generating, isolating, and analyzing monoclonals have come a long way, and we're seeing that during COVID-19. It's month 10 of the pandemic, and we're already getting some efficacy data in humans. And compared to the past, that's incredibly fast. Still, monoclonals also face challenges in the time delay to get to scale. As they're currently manufactured with mammalian cells, it takes many months to go from the seed to high-scale manufacturing. But it's possible that different production approaches, using perhaps fungal cells or plants, we could see a 5-fold reduction in that production lead time. And there may even be different ways to produce monoclonal that are cheaper and faster. Nucleic delivery platforms have the potential for accelerating response times even more. And this is an approach I'm personally quite excited about because it would eliminate that step of having to make the antibody proteins and instead just use the body, given the genetic sequence, to make the antibodies itself. And so once you're dosed, would become the monoclonal factory. A third area that we need much better innovation in is diagnostic platforms. COVID, because of the asymptomatic transmission, has shown us that if we want to block change of infection, we can't just look at the people who are symptomatic. In the United States, 3 out of 4 people infected with COVID-19 are never diagnosed. Usually that's because they either have mild symptoms or none at all. And so given the difficulty of getting to testing and the limited scale we have with current techniques, these people are never diagnosed, and so they don't change their behavior and reduce the spread. Even sometimes when people are tested, the results come back negative because some of the tests are not sensitive to small amount of virus. But of course, those people often, as the virus grows, they infect people. So the diagnostics are letting us down. Right now, the current business model is based on finding people with symptoms, and we need to change that. We need sensitive and specific diagnostic for asymptomatics, and we need to make it easy to access. The good news is that the technical piece, the PCR testing, is sensitive and specific. PCR finds that needle in the haystack. A lot of our challenge, though, is the logistics. One vision is that we should have a test kit that we can spread out into the community, either into medicine cabinets or community centers, pharmacies. It would just be a Q-tip swab and an envelope. And anyone who was exposed could self-diagnose without having to stick it too deep into their nose, and then they'd mail off that envelope. Lots of workplaces, airports would have this, and we need to get those PCR machines then using a reel-to-reel process that can process 100 times as many samples as the current best that only do a few thousand a day. Another approach is to use a point-of-care diagnostic platform called Lemura. It's small enough and also very sensitive and can get out to primary care. Also, it's even portable, so a health worker might be able to bring it into the community itself. So diagnostics is another key area that we need to be far better next time around. I'm optimistic that given the scale of this pandemic, although we weren't ready this time, we'll learn from our mistakes and do far better in the future. I'm optimistic about this because so many of you are working at the forefront of the areas I've talked about, and that includes vaccines, diagnostics, and therapeutics. For this whole 16 years, the Grand Challenge community has joined forces to apply the best science to the world's most important problems. And I think if we continue to push science forward in the ways I've described, we'll not only solve the world's great challenges, current challenge, the pandemic, but we'll also help with other diseases, and we'll be ready for the next one. With that, let me go ahead and now introduce our keynote speaker. Before COVID, of course, we'd planned for this meeting to take place in India. And India's very inspiring because it's made huge strides in improving the health of its people over the past 2 decades. And now, India's research and manufacturing capacity will be critical to fighting COVID-19, especially when it comes to making vaccines at large scale. To speak more about the country's role in advancing health R&D, I'm pleased to introduce Prime Minister Modi. Welcome to Grand Challenges, Prime Minister. Our foundation is grateful to partner with your government to improve public health in India and around the world. We look forward to what you have to say.